Okay, hello. Now for part 2 of coronavirus replication. So, shown here is from the famous Eric Schneider paper, and he's the guy that wrote this review that we like. And once again, the link is right there on the side. You can go get all the words, instead of just some of the words. So this is the replication factory of a coronavirus. And so you can see it's got all these different parts. It's got big double membrane vesicles out there. That's our DMVs. It's got little open pouches that he calls double membrane spherules, S-P-H-E-R-U-L-E. Yeah, spherules. That's fine, yeah. And more importantly, this is the paper where they discovered the pores. And so here we can see the pore coming out of the double membrane vesicle. And you can see at the double membrane vesicle pore, and there we go. And this will be sort of RNA lined up on the inside, and it'll be coming out to the outside, something like that. And the double membrane vesicle pores, we believe, are made from NSP, which is non-structural protein, which means part of polyprotein 1A and 1B. They're made from NSP 3, 4, and 6. 3, 4, and 6 are believed to all be in there. And they are not just membrane spanning. They cross this membrane and they cross the other membrane. And they staple those two membranes together to make a pore. It's just a nice sort of rigid structure that goes through the membranes. And lets the right things in and lets the right things out for the coronavirus perspective anyway. Okay, so how do we go about doing this? Well, the first step in coronavirus replication is for the virus to recognize its own RNA, because the cell is a complicated place, and it's got ribosomal RNAs and tRNAs and lots of messenger RNAs swirling around all the time. Tons of RNA, far more RNA than DNA inside of a cell. So the coronavirus needs to be able to recognize its own RNA, because when it replicates, that's the one thing that matters to the virus, basically. Yeah, we're humanizing it a little bit, but I think that helps. It's not a terrible thing. You don't necessarily put that in a test answer, but that's how I see it anyhow. So how does the virus recognize itself? Well, it folds up and it makes a shape. So here we have a series of what they call stem loops, or you can call them hairpins. And this is just RNA from the virus that's folded up with everything on this side base paired to everything on this side. And where you see a bulge, that's where you have nucleotides that they're not base paired, and you might have different numbers of them, something like that. And so, the 5 prime, which is at the beginning of the genome, untranslated region, UTR, is going to fold up into a shape roughly like this. And we know that we have that signal, and we also have a structure down here in the 3' untranslated region, where we have a big, long, bulgy stem loop like that. And we also have a thing called a pseudoknot, where this little loop can fold down and actually base pair right down here. It's a little tough to draw in sort of 2 dimensions like they've done here, but it's a little pseudoknot structure. And also, on top of that, the structure right here, about where the pseudoknot is able to base pair with the structures right here. So stem loop 3 can either be together, or it can unwind and base pair with the tail end of the virus RNA, the 3' end. And so when it does that, the virus has made a circle. So it's got its 5' end, it wraps around, and then the 3' end actually binds to the 5' end. And so we call this structure a panhandle, P-A-N-H-A-N-D-L-E. Just like it's a skillet or something. It's got a sort of a handle on it, and then it's got a big round part, and that's the genome that's making a big loop. And so they've just drawn it as this, but the genome is enormous, and so really, it's a really big pan with a really small handle. Yeah. A lot of viruses form 5' and 3' and sort of panhandle structures. And this is used in replicating the genome because when you have a panhandle, you have a nucleotide that you can basically use as a primer, and it's bound to another nucleotide that you can basically use as a template. So it's like a way that you can potentially, generate, get things started in terms of replication. All right, what's the process? Here's the process. See the process? Love the process. Look at all those processes in the process. All right, once the virus is inside the cell, and once it is translated and made some replication proteins, so once it has made a couple of copies of open reading frame 1AB and translated polyprotein 1AB, then it has the enzymes to do all of this stuff. And so those are going to first take the initial strand. So our virus starts out, and we call the strand that comes in the positive strand in this virus because it's in the right orientation to be translated directly. And we're going to make a copy of that. So a positive strand is going to have a cap on the 5' end and a poly A tail on the 3' end, just like a messenger RNA. That's how it gets translated. Now, if you were to make a copy of this, remember that the two strands in any RNA or DNA hybrid go in opposite directions. So if the 3' end is here, then the copy of it would have a 5' end that would start with U, 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 wouldn't have a cap because it doesn't get capped, and then would go down here. And instead of a poly A tail, it would have whatever, it writes from this region. And the polymerase can't actually copy the cap because it's a backwards nucleotide and it's not able to be copied. So that strand with a short poly U in play at the 5' end instead of a 3' poly A is called the minus strand because it doesn't have a cap and it doesn't have a poly A tail, and even if you could feed it into a ribosome, it would encode nothing because all the genes are reverse complement. They're backwards and upside down, essentially. So it's just nonsense. But the minus strand is there so that the minus strand can be used as a template. We're going to make copies of the minus strand, and those will be new positive strands. And so the virus's purpose in doing this is to make a minus strand, and from that minus strand it can start to run off thousands and thousands of copies of positive strands. Now the virus does two things while it's making minus strands. One, it can just make a minus strand all by itself, and that's great. Or number two, the virus can, as it's making the minus strand, read these sequences. So these sequences are called transcription regulatory sequences. And there's one right at the front of the genome, or near the front. It's about 70 nucleotides in, or 75 nucleotides in, they say here. And then there are several throughout the genome. I think SARS-CoV-2 has, it's got 8 of these in the body and one of these at the front. Yeah. Each time, and they all have the same sequence, so each time we're copying the negative strand, as we read one of these, there is a chance that the polymerase is going to stop and say, you know what, I've done enough here. I'm going to finish this up. We're going to wrap this up. And so what it does is it pauses at the body TRS and it transfers up to the leader. Now it just takes the body TRS, which base paired, or it takes its copy of the body TRS. And since the body TRS and the leader TRS have the same sequence, it knows that this will still base pair up at the beginning. And so you take this, move it up there, and then complete the strand. And so in this way, the virus will make a much shorter version of its RNA. And it'll have the complete 3' end, and it'll have the complete 5' end, but it'll be missing some chunk from the middle. And so you'll make what they call a nested set, Nested, like Matryoshka, the little Russian stacking dolls, where the little one is inside the slightly bigger one, etc., like that. And so they all have the same beginning, they all have the same end, but they have more and more of the middle. And so you can look at the genome as like the biggest one of the little stacking dolls. Yeah, this one would be the smallest one that only has one body TRS in it. And this is done at the point where negative strands are made. So the way I think of this, first we get to a cell, and the polymerase is, think of it as like a kid. It's young, it's easily distracted. And so when the polymerase sees a TRS, a transcription regulatory sequence, it's just like, wow. Like, oh, and it stops, it pauses, it sort of, thinks about things. And then a lot of the time it just can't go on. It's something like, I don't know, 25% of the time it will stop and transfer to the leader, and the other 75% of the time it'll keep going. And so in this way, every time you stop here, a portion of your polymerases will terminate and will move up to the leader, and then will complete a little subgenomic RNA like that. That's shorter than the genome. And so you'll end up with the most copies of the smallest one, because that's the first thing that every single polymerase sees. And so you get the full 20% or whatever. By the second one, you've already subtracted 20%. They already went up and made one. But of the ones that remain, you're going to lose another 20% at the next one. And you'll lose another 20% at the next one. And so you end up with this sort of gradient with lots of the smallest one, and just sort of steadily less and less and less at each larger RNA species. And you actually don't get very much of the intact genome. So the intact genome is what you get only if the young and easily distracted negative strand polymerase is able to skip all the tempting TRSs. Once the polymerase gets to the end and it's made a full negative strand, something changes. And we're not sure exactly what. We think it involves both the host cell and maybe changes to the virus. When that changes, then the polymerase is going to switch. And from that point, instead of copying a plus strand to make a minus strand and maybe getting distracted along the way, it's going to never make minus strands again. It will only make plus strands, and it makes them very efficiently, and it does no skipping. So we copy each of these minus strands, however big they are, including the entire minus strand genome, and the plus strand, the late polymerase, like the older, very work-focused polymerase, if you like, just plows through. It plows through whatever it sees, doesn't stop at any of the TRSs, doesn't do any jumping around, and it makes lots of copies. And so the little subgenomic RNAs that are positive stranded are going to get a 5' cap, and they're going to have a 3' poly A tail, just like messenger RNAs. And so those will go off and be translated to make all the other proteins that the virus needs. It made its polymerase and all its replication enzymes from open reading frame 1, and it didn't have to copy anything to do that. That just came in on the genome. But to make things like the spike and the E protein and the M protein and the N protein and all the other little proteins, it's got to make subgenomic RNAs. And from each of these subgenomic RNAs, even if it has, like this one has 1, 2, 3 genes on it, only the first gene ever gets translated in any abundance. And so this one will be used to translate this gene, this one will be used to translate this gene, and this one will be used to translate this gene. Yeah. First gene after the TRS gets all the love, the other genes get none of the love. That's just how it is. Yeah. I don't make the rules. All right, so that's fine. That's good enough for this section. And then we'll get into the actual mechanics of how the heck does the virus do this in the next section. So stick around, or I don't know, go get a drink or something. Yeah, then come back and your brain will be focused. All right, thanks very much.